So if you take a look in one cell, there will be proteins, there will be DNA, there can be RNA too. So which of these DNA, RNA, proteins will become genetic material was in question. So they started to search what is the genetic material. In 1928, Frederick Griffith did experiment with mouse and a bacteria called Streptococcus pneumoniae. Streptococcus pneumoniae. Streptococcus pneumoniae are available in two varieties. If the Streptococcus pneumonia, if they are not covered with any polysaccharide coating, then they are called as rough strains. Rough strains. And they are harmless. Whereas there are Streptococcus pneumoniae that are covered with capsules. They are covered with a coating and they appear smooth in the microscope. So they are called smooth strains. The smooth strains are virulent. They are harmful, whereas rough strains are non-virulent. So what Frederick Griffith did is that he introduced the rough strain of bacteria into a mice. And since the rough strain is harmless, the mice was alive. And then he injected S strain of Streptococcus pneumoniae into mice. The mice became affected with the pneumonia and the mice died, which proves that S strains are harmful for the mice. Next, what was done is they used heat-killed S strain, and that was injected into mice. When heat-killed S strain was injected into mice, the mice were still alive. That means heat can kill the bacteria, and it did not affect the mice, so the mice was alive. After this, so these are the three steps first he used. So there's one R strain and then S strain. Then third is heat-killed S strain. After that, what was done is he mixed the heat-killed S strain was mixed. Heat-killed S strain was mixed with live R strain. So R strain is harmless. And then when this mixture was introduced into mice, what was found is that the mice died. Why did the mice die? What happened is that there was some factor that is present inside the heat-killed S strain. That factor did not die even after the heat. And from the heat-killed S strain, that factor migrated to the body of live R strain, transforming the R strain into S. So R strain, which was harmless, became harmful. It got converted to S strain, and so when it was injected to mice, the mice died. So this proves that there was some transforming principle which was present in the S strain. There was some transforming principle which is present in the S strain, that even after it is killed by heat, that bacteria was killed by heat, but that factor did not die by heat. So that factor, whatever was present in the S strain, transformed the R strain into S strain. So the R strain got converted to harmful S strain, and that created disease for the mice, and the mice died.